And then I go to
SynBio in a search for life in the universe. So allow me to introduce Lynn to you. She's an astrobiologist and synthetic biologist at NASA's Ames Research Center, and is also an adjunct professor at Brown University. She's interested in the origin and evolution of life on Earth and in the universe. Presently, she works on a thesis of synthetic biology to enable space exploration. Her astrobiology research has taken her into exciting field sites, such as the Yellowstone National Park in Kenya's Rift Valley. She appears frequently on radio and television programs. Perhaps you have seen her appearance in Morgan Freeman's Through the Wormhole. So Lynn definitely has a long list of awards and accolades. I won't list them all tonight. Just to share a few, in 2015 she was awarded the Isaac Asimov Award from the American Humanities Association, and has been a NASA Innovative Advanced Concepts Fellow 3 times, most recently in 2018. So we're very honored and pleased to have you here with us this evening. Can we put our hands together to welcome Lynn?
Thank you very much. I'm always impressed when people come out on a cold night, particularly in the middle of the week. A couple of years ago, I was packed during exam time when I gave a talk in Norway, and I discovered it's because they had bribed the students with Mars bars and herring. So I don't know what they bribed you with tonight, but thank you very much for providing it. Anyway, I know many of you have had a long day of classes and studying and so on, so hopefully this is going to be a lot more fun than maybe you've had earlier in the day. And I'll give you a little bit of a taste of some of the things that I've done since I've been at NASA. I certainly didn't expect that I would be spending my career there, but I have, and it's been quite a ride. It's very different from being in an academic setting, although I run a large biology lab that would look to all of you more like a regular academic lab with students and postdocs and all that sort of thing. So with that introduction, and to say, in spite of what you may have read on Twitter, which is almost as reliable as Wikipedia, I suppose, I am not an astronaut because I'm too big a coward. But I do know a lot of astronauts, and I have met Neil Armstrong, and I have met Buzz Aldrin, and Jack Schmitt, and Rusty Schweickart, and a few of the shuttle ones. So if anyone wants to ask me afterwards about that, I'm more than happy to tell you. But sadly, that's the closest I've gotten to actually becoming an astronaut. So anyone who wants to leave now, quietly leave. And for the rest of you, can we turn the lights down a little bit? Because I think the slides will be a bit better. And I'll trust that all of you stay wide awake. This takes a PhD. There's a switch that might work. It takes a gun and shoots into a light bulb. Oh, brilliant. Yes, it's very high tech. So, I would argue that since the dawn of humans, we've wondered, are we alone? Our ancestors, we know, looked up at the sky and saw what they categorized as constellations and so on. And we know that they thought about what might be out there. I mean, it's one of those questions that I believe is deeply embedded in us, sort of the sort of questions that you ask once you figure out how to make babies and what's for lunch and so on. It's something that's very much ingrained in us. But what's interesting is this is the very first time in all of human history that we actually have the scientific tools to be able to ask these questions and come up with some answers. So I'm going to start off by talking a little bit how we might look for life in the universe. And to do that, you start with Earth, and then you ask questions like, could it happen again, and where, and how will we know? So I said that this is the first time in human history that we've been able to really tackle these questions scientifically. And to me, it really boils down to about 4 different things. First, we have space missions now that allow us to go not just to the other planets and the moons, but even beyond the solar system. So we've got our robotic surrogates out there actually interrogating other bodies in our solar system and beyond. We know about extrasolar planets. I don't need to tell this audience about the latest Nobel Prize, I'm sure. And wow, isn't that great to be able to get a Nobel Prize for your PhD thesis? I wish I'd thought of that. And so yes, we now know about many, many extrasolar planets. We know much more about life in extreme environments, and I'll talk a little bit about that. We know much more about molecular evolution. So when I was a graduate student a gazillion years ago in the last millennium, I was studying protozoa and algae, and one of the big things that we used to look at phylogeny was the structure, the ultrastructure of the flagellum. You know, is this significant that it has 9 plus 2 or 9 plus 0 or 9 plus 3? And what about the subcortical alveoli and so on? And you puzzle over these micrographs and so on, and you argue vociferously late into the night. But at the end of the night, sort of the conclusion was, I'm right, you're wrong, but we will never know for sure. But now with the molecular techniques, we know far more with a much greater level of certainty of how evolution works. And I'm going to argue and throw this in that synthetic biology is sort of the 5th leg of why this is a great time to actually be looking for life in the universe. And for those of you who weren't expecting me to sneak this in, synthetic biology, and it's kind of a fun building to say this in, is really the design and construction of new biological functions and systems not found in nature. This is pretty much what came out of the British government and it's been adopted by the US government. And we could make an analogy with synthetic chemistry that first you do descriptive chemistry and then we moved on to synthetic chemistry 100 or so years ago. And we're now doing the same thing with biology. But this is probably as good a way to describe it as any. If you don't laugh, you're not awake. And I also want to slip in because a lot of the projects I'm going to show you are the result of an iGEM team that has been in my lab since 2011. And some of you now may not be aware of the iGEM competition. This is the International Genetically Engineered Machine Competition. It's primarily undergraduates, but there are some graduate teams, there are some high school teams, and what's kind of fun doing this here is that the Cambridge team from maybe 2010, 2009 won the whole thing. And their project is still famous for E. chromide. Jim Hazeloff led that group to victory, but I know there hasn't been a team for a couple of years. He's sort of on to other things. But Cambridge is very famous in the iGEM lore. But what happened is in 2011, through the fact that at the time I was teaching at Stanford as well as Brown, I was asked to become a faculty advisor for an iGEM team. And I thought it was sort of a stopgap measure, and I say I've been doing this since 2011. But since my lab is at NASA and I'm paid by the US government to do space type things, obviously we had to have projects that were focusing on space. So we started in 2011 tackling the question of could you use synthetic biology to enable a human Mars colony? And then we went on to astrobiology and using synthetic biology to answer some of those questions, which I'll just touch on later today. Then we did something with de-extinction and a couple of other things that you'll see a little bit of later, thus the dinosaur in the spacesuit, a biodegradable drone that I'll show you a little bit later, self-folding origami, which was great fun, but I will not show you, a bio-balloon, which also I will not show you, some various projects. This gets longer each year because we keep going. Mycotecture, which I will show you a little bit, and the astropharmacy, which I will show you a little bit, which we just finished competing with. The great thing about the adjunct competition is all the projects require a wiki. So if you're interested in any of these projects, or the Cambridge ones over the years, please look at our wiki, as I tell the students all summer to say. The answer to everything is, please look at our wiki. But let's get back to the main show. So that was just sort of background so you know what's coming. And that was the question of how you look for life in the universe. And I sort of distilled this down to 4 or 5 steps, which makes it a whole lot easier for you, because then you can just take quick notes and you're all set to go be a Martian hunter. So the first thing, if you're hunting for anything, is you have to know what you're looking for. And if you're out hunting for a deer or a lion, you know what a deer or a lion looks like. That's no problem. But what if you're looking for a life form and you have no idea what it's going to look like? So what can we hypothesize about life? Now, many people just simply extrapolate from life on Earth, and they say, well, all life on Earth uses DNA and RNA and proteins and this and that, so we must have to look for that. But is that the only way you could make life? In fact, is there even a definition for life? And the problem is, as I'm sort of alluding to, we only have one data point. So when I got to NASA in 1987, I actually looked down on some of the astronomers. I thought, well, you know, you have to wait to get data until there's another mission and so on, and I can go into the lab every day and do useful things. And at that point, we were pretty much even. We knew about one solar system, we knew about one life form, you know, draw. But as of a few nights ago, before I left town, 16th of November, this is where we were. 3,039 solar systems and 4,093 confirmed planets, extrasolar planets. 161 are terrestrial-like, so somewhat like the Earth in that they're rocky with iron-rich cores, and still, guess what? One life form. So, I cannot tell you how thrilled I would be if I could stand up here and say that there's even one other life form that we found. You know, I would be happy, much less 4,000 other life forms, because then you could say 73% use DNA and 22% have lipid membranes and so on, and then you could get some handle on this. But we don't have this, so we're going to have to sort of hypothesize. So we ask things like, do you need to use carbon as your basis? And I would argue that yes, we do, for all sorts of reasons that I can't go into tonight. So those of you who are here for a silicon-based, you know, life talk, I think there are a lot of good reasons that you're not going to find it, certainly not nitrogen. In fact, you know, what is life? And I think this sort of boils down to some extent to a philosophical point. So it turns out that Da Vinci wrote what life was. He said, water. Water is sometimes sharp and sometimes strong, and it's sometimes acid and sometimes bitter, sometimes sweet and sometimes thick or thin, and so on and so forth. You know, it sometimes brings hurt or pestilence, sometimes, you know, life-giving, and so on. All these things that obviously, if your professor asked you to tell him what water was or her, you would not be expected to write this. You would be expected to write that, right? That's a natural definition. So the question is, do we actually have a natural definition for life? And after thinking about this for many years, I have a very bad feeling that there is no such thing as a natural definition for life. And so if that is actually true, since we know life is historical, so molecules are interchangeable, but organisms aren't, and if life is a non-natural kind, like I'm suggesting it is, that means that some other life form may not define us as alive. So that's kind of a little bit of a scary thought. But you have to do something. You can't just tell the administrator at NASA, I give up because I can't define life. And so I start by saying, well, I'm going to assume that life has to have some sort of physical instantiation. It's not just a computer game. And that it's going to be based on organic carbon, and that's because carbon is certainly the most flexible element for making all sorts of compounds. We see these in the interstellar medium. We even see things like amino acids. We see the language of life. And so sort of the scary thing to me is, I don't know how many of you have suffered through biochemistry, I did, and then ended up getting a PhD in it. But there are some people who are gluttons for punishment. But anyway, you see these molecules that life on Earth uses, even in the interstellar medium, so you wonder, are there students around Alpha Centauri, for example, who are taking biochemistry, who use basically the same textbook that you use, except an Alpha Centaurian? I mean, maybe they're hacking into your websites, I don't know. But maybe there is really a language for life in a broad outline. And I think in the broadest outline, you're always going to use amino acids and you're going to use organic carbon because it's out there, it's all over the place, it's easy to make. And I think sort of the final kind of fun argument is that if you want a place for silicon-based life, this is the perfect one. Planet Earth is a large silicate rock. There is almost no carbon on planet Earth. It's something like 0.03%, but we've got a very large percent silicate. And yet we are not based on silicon. So this would be a perfect place to have a silicon-based life form. And even with all that, we are carbon-based. So again, this is leaving you with a little bit of a creepy feeling that maybe life is not a natural definition, but it's something with organic carbon, and let's play a little bit with that. So we can get some idea, even though we only have one data point, from looking at life on Earth. Now I realize that many of you are in the sciences, if not all of you, and it's very, very difficult to publish a paper with one data point. But if that's all you've got, you're going to have to extract whatever information you can. And so one of the things we can do is look at the range of life on Earth and get an idea where it lives, so at least we have some idea where it's possible to live elsewhere. And so what we do is we look at organisms that live in extreme environments, and people call these extremophiles. They are not a taxonomic group. There are extremophiles all over the phylogenetic spectrum, so it's more like saying all organisms who are blue. It's really just a group that has a particular characteristic but not phylogenetic. So I love showing this picture. This is one of our field sites in the Rift Valley in Kenya. The reason you go to the Rift Valley is it's got some of the highest pH lakes in the world. But it also is a great place to show a lot of other environmental extremes. For example, you can see the boiling water behind me, very high temperature. We know that there are organisms that can be metabolically active from about minus 17. The Himalayan midge is reputed to be active at minus 17, all the way up to about 121, 122 centigrade. That's a huge range. I mean, it isn't if you're an astronomer, it is if you're a biologist. And we're not even including things like spores that can deal with boiling water, or organisms that can go into a spore sort of form. And you think, wow, that's amazing. And then I'd love to point out that there are things called, for example, penguins. Many of you probably heard of them, that live in the Antarctic. They are metabolically active well below minus 20. And I try not to mention this when I'm being recorded, but I did give a talk at a relatively prestigious university in California once and pointed that out, and a professor immediately said, but they don't count, they're cheating, they've got feathers. Ladies and gentlemen, there is no rule book that says if you have feathers or fur or fat, you don't count. Or I mean, the fact is that they are metabolically active well below minus 20. Those are perfectly good adaptations. So they're not cheating. So the answer is that the range for life, for survival, is far greater than this minus 17 to 122. And when you start growing fat or fur or feathers or whatever, you can also extend that stretch. What is very difficult is to go to the very high temperatures and be metabolically active because, as I'm sure you're well aware, your proteins and your lipids and your DNA and so on are all in nature at certain temperatures. So pH is another sort of interesting one. Most of us live in that sort of pH 70 world. The oceans are about, well, we used to be a little over pH 8, slightly under now. But we know of organisms that literally can go down to pH 0 and up to 11 or 12 in places like the Rift Valley. You're going to places like Yellowstone to get the ones down around 0. And if you think hard about your chemistry, you actually can go below pH 0. Part of the problem has been that people don't sell portable meters. They're starting to for that sort of thing. I do have colleagues who insist that they've seen things below pH 0. I'm not sure if it's been confirmed or not yet. So this will, probably even broaden a little bit more. So this is an enormous range for life. Desiccation, if we're carbon-based but we use water as a solvent, obviously lack of water is going to be an extreme environment. And so we do know that there are organisms that can undergo anhydrobiosis. They can get rid of most of their water, 75% or so, and survive for long periods of time, and then come back to life. But in terms of actually being able to metabolize, we really can't deal with too much loss of water. And sort of along those lines, salinity. You need a little salt to have your chemical reactions go, but too much salt also creates a problem. So there are organisms that are able to metabolize very slowly, we showed over the years, in solid salt. But they're not ripping through, but they are able to at least metabolize. So you need some, they do all sorts of tricks like making compatible solutes and so on, but it is possible. Various chemical extremes, you read things now and again when someone proposes organisms and things like benzene, different pressures. There's some organisms that live in the bottom of the ocean, you bring them up to the surface and it's like, oh, this is what it's like up here. And others that you bring up to the surface and they explode. So there's obviously what's you're not seeing these things that are popping up on my screen. Radiation, that one that's been near and dear to my heart because that's particularly important as you go past the cocoon of the Earth. We're exposed not just to ultraviolet radiation at shorter wavelengths than on planet Earth, but then galactic cosmic radiation and all sorts of nasty things, electrons and protons and all sorts of things that you wouldn't want to be exposed to. And so this is something that's a very old stressor and becomes, again, very relevant as we go beyond planet Earth. And sort of along with that is oxygen. You think, wait a minute, oxygen, that's crazy. We're living in a 21% oxygen environment. It may even be that high in here. But living with oxygen is really a dangerous game. You're literally playing with fire. You're burning things. So oxygen isn't so bad. Water at the other end isn't so bad. It's all these reactive oxygen species in the middle that you're producing that create oxidative damage. And I know there was some period in the literature that it seemed like there was a paper for virtually every ailment known to mankind that blamed oxidative damage, from preeclampsia and premature birth to cataracts and corneas. And this is, it's like the world, all the ills in the world were because of oxidative damage. May have calmed down a little bit. But it makes you sort of wonder then, are we extremophiles too? Because we're actually very well suited to live in 21% oxygen. In fact, I would argue that if we weren't aerobic creatures, the graduation requirements at Cambridge would have to be vastly lowered because you wouldn't be able to support these huge drains and this high level of metabolic activity. So I would argue that we are actually extremophiles too, we just don't think about it because we do that. That's part of being an animal. So now you've got some idea of these parameters, at least in a linear sense, that life can live on the Earth. So if you start putting together, yes, we know of organisms that can live in basically boiling battery acid in Yellowstone. So they're down at around pH 1 or 2 in boiling water. So we can start to fill in this niche space for life. And what that allows us to do is to go elsewhere and say, here's a place on Venus, or in the past on Venus, where you have boiling battery acid. Because we know of organisms in Yellowstone that can live under those conditions, it means it's at least worth looking there. It doesn't mean that the same organisms are there, it just means that we know it's possible for an organism, even in our limited one data point, to live there. So we start to look elsewhere. And one of the nice shorthands is to start with this desiccation. So you check where there might be water. And so obviously Mercury is a non-starter. So you start to look at some place like Venus. Now, Venus is miserable today. In fact, the word hellish is often associated with Venus. But Venus is very close to the Earth. It's very much Earth's twin. And of course, what happens to the sun is, as every star, it burns, it becomes hotter and hotter. And so Venus at some point hit a tipping point and had a runaway greenhouse. So the carbonates went up into the atmosphere, so you have the carbon dioxide creating a greenhouse effect, and ultimately the water also then goes up in the atmosphere. So you have a runaway greenhouse, and this is what will someday happen to us if we don't do something about it. And so this was not a good scene. In fact, this may have actually only happened about 500 million years ago. And we know that there was a lot of life on the Earth then. In fact, life's been around for several billion years, at least 3.5, probably more. And so if life was present on the Earth, and the same conditions were present on Venus, it's at least possible that life also arose on Venus. And in fact, it may even be probable if life arises under certain chemical and physical conditions. And even more than that, it's quite possible that there are meteorites and so on that went from Earth to Venus just the way we have ones from Mars here. So let's say that there had been life on Venus and it got hotter and hotter, what would you do? So one thought that Carl Sagan actually proposed in the 60s was that life could have ended up going up into the clouds, because if you go about 60 kilometers up, it's more like a pleasant day in Cambridge. They're organics, it's in the 20s and so on. And so it's possible there's life up there. One of the problems with this idea is if you go outside, you don't see green or orange or pink clouds here, do you? So we know there are organisms up there in the clouds, but not that many. And the question is always, are they in transit or is there really an ecosystem? And periodically I've seen articles suggesting there's an ecosystem. But honestly, these are people who stood up in mountains in Switzerland and grabbed a sample. So yes, you're X number of meters above sea level, but you're still only 2 meters above the ground. So I think that the jury's still out on that. And so our 2012 adjunct team actually did some work along those lines, creating organisms that could tell us that they were dividing so that we could put them in a cloud chamber. But didn't get as far as actually getting a cloud chamber. So Venus is a suspect. Worth looking, pretty close, relatively cheap to go there. I mean, we're not taking up a collection at the end of the talk, but the grand scheme of things, it's cheaper than a lot of things. Planet Earth, we know it's infested with life. If you don't believe me, look around the room. And so then we move merrily towards Mars. Mars is obviously the opposite situation from Venus, in that it's a little farther from the sun, but that's not the real reason it's so cold. What happened is, instead of a runaway greenhouse, it's smaller, it lost its tectonic activity, it doesn't have a gravitational pull, so it lost its atmosphere. So the thought is that earlier on, it was also much more similar to the Earth because it had much more atmosphere. And so we think that there's evidence of dried lake beds on Mars. The Mariner missions in the 60s showed those. I used to show pictures of them that reminded me of black and white televisions at the time. And by golly, they look like dried lake beds and river beds, so they've been interpreted that way over the years. Obviously, we have much more information about Mars now. About every 2 years, there's a great time slot to have a mission to Mars, and so we've really been on top of it. We, not just NASA, but the different space agencies, European Space Agency, and various others, trying to take advantage of these windows. About every 2 years. It takes about 6 months then to get there. So right now we've got a couple of things going on the surface. Europeans have got things that they're sending, their orbiters and so on. Now and again we get reports that Mars is belching methane, so you're thinking, well, maybe there are organisms beneath the surface. There could still be liquid water beneath the surface, even though it's below the triple point on the surface, so you can't get liquid water right on the surface. So, it's still suspect, and again, in the grand scheme of things, it's fairly cheap, and actually it's a better place to go than Venus because, as I say, the surface of Venus is hellish. It is the hottest surface in the solar system. It's even hotter than Mercury. So the story has it that the Russians, had a couple of missions there, actually when it was the Soviet Union, to the surface of Venus, and that at least one of them, and this may be apocryphal, but I love the story, included a little lead bust of Lenin. And so the story is there is now a little lead pool, formerly a bust of Lenin, somewhere under Venus. Whether that counts as life on Venus, bad. Anyway, so Mars, a lot more pleasant place to land, and so on, and we are on it. Ceres. I'm always going to say, be serious. Ceres is the largest object in the asteroid belt. It's got more mass than everything else put together. And now it looks like there's ice on it, water ice. And, then could there be liquid water beneath the surface? And you start to think all these crazy things that we never used to think of before. Europa, this moon of Jupiter, Jupiter, I'm sure everyone in the room's heard of it. We know that it's ice-covered, very high albedo, very bright white. You know, it's amazing. Some of these things are not actually that complicated to figure out. Very bright white. We know from magnetometer data from Galileo that there's a liquid water ocean. It's got a certain salinity. It stays liquid because of tidal flexing with Jupiter. You know, something that we hadn't thought about several decades ago. You know, you think, how could you end up with liquid water beneath an icy crust that far away from the sun? Well, it's the tidal flexing. And on top of it, there's some discoloration. And we think that that involves organics. And there have been all sorts of ideas on how you could run an actual ecosystem beneath the surface. We know that icy crust has a lot of movement. Maybe you could get organics made from all the radiation coming from Jupiter onto the surface. You get the grinding between the plates. You get some of this material going down. You know, maybe that's how it works. Now, obviously, I would love to have a penetrator mission to Europa. I think many of us would. And we did have a big champion in Congress for a while. Unfortunately, he was not reelected. So we're trying to salvage some of these, you know, even an orbiter or a lander would be great. But Europa certainly should be having much more exploration. They also apparently periodically has geysers, which would be great if you could count on them, because then you actually can probe the interior without having to drill in. And so, you know, again, Europa is fantastic. Europa also is roughly the same size as the moon, our moon. And as you probably know, the moon is going away from the Earth at about 4 centimeters a year. It was actually discovered by one of Darwin's sons, who, you know, the college down the street and all that. So I suggested that if we're going to lose our moon, we could bring Europa in, because then it would be a whole lot cheaper to study. It would only take us like 4 days to go there. I've been saying this for over 2 decades, and I'm getting no traction whatsoever. But I'm sure it's just a matter of finding the right multi-billionaire, and we've got it. Obviously, there's some other moons of Jupiter that are interesting, Ganymede and so on. But let's move merrily on to Enceladus, which is a tiny little moon of Saturn, and it's a similar sort of situation with the ice-covered liquid water interior, and even better because there are about 110 or so geysers in the southern hemisphere, called Cold Faithful, an homage to Old Faithful, and it's like having a spigot into the interior. And so the Cassini spacecraft, which was orbiting the Saturnian system for quite a while, was not there to interrogate Enceladus. In fact, we didn't even know that there would be a liquid water ocean and so on. But Cassini sniffed oxygen. Turns out it was because the water was coming out and was breaking apart. And anyway, the long and the short is, in spite of the fact that this mission wasn't designed to interrogate Enceladus, they found all sorts of organics coming out, the sort of things that would make people in this building, you know, feel warm and fuzzy. And so obviously we would love to have a mission back there. And there are actually talks not just about NASA, but some private missions to some of these suspect places as well. You know, unfortunately, it takes a while to get there. And just to be clear, it is Enceladus, it's not enchilada. I have heard of I'm sorry, I left out there. Obviously, is another very interesting moon of Saturn called Titan, which is like a frozen chemistry experiment. And I love that because you see these pictures on the surface, and it looks so ordinary. I don't know if any of you have ever flown into Chicago, but it always reminds me of pictures of O'Hare as you're coming over the lake. And some days when I'm feeling particularly evil, I even taken pictures landing in Chicago, pixelated them, and turned them black and white, and pawned them off as pictures of Titan. I did that at Berkeley once, and I said, you know, and of course we all remember when, you know, we got those first images of the Huygens probe of the surface of Titan, and people were, oh yes, I read it, so excited. But I don't want to give them too much myself. But anyway, it's a frozen chemistry experiment. The liquid is small organics, methane and ethane and so on, and the rocks there are frozen water. And so you start to think, could there be an alternate chemistry, something sort of inside out maybe, where you had the, you know, an aqueous layer, you know, or silicate or whatever with organics on the inside, whatever, or organics as a solvent. But then it turns out some of the newer data suggests that there actually is liquid water beneath the surface. And so now you're like, everything is aligned. You've got excuse the expression, a home run, because you've got tons of organics there. And if you've got the liquid and so on, source of energy, it's, obviously be a really cool place to go back to. So, in fact, NASA's just selected a mission to Titan called Dragonfly, which is kind of cool because it's a drone mission, which is really cool. So the problem with sending balloons or drones to a lot of these places is the atmosphere is really thin. So like Mars has got 95% CO2, but it's only 7 millibars, and the atmospheric pressure on Earth is 1 bar, so you're less than 100th. And so it's very difficult to get the lift. But you go to some place like Titan or Venus. Venus is hot and miserable, but there's at least a lot of atmosphere. And so ballooning, at least in the clouds above Venus, is not unreasonable. And Titan is really the one other place you can pull off something like Dragonfly or a balloon mission. So that's going to be really exciting. One of my colleagues is involved with that. So anyway, and I won't bore you with that former planet called Pluto, but no self-respecting astrobiologist would even discuss Pluto until New Horizons, or a few months before, when the suggestion started to be floated that there was ice on the surface. And now these trans-Neptunian objects. And now we know there are other ways to create liquids beneath the surface. You're very hesitant to say no, because it's really now maybe. And so the reason I show you all these places is, golly, we now have how many suspects? 10 or more in our own solar system alone, where there could be independent origins of life, or life now that traveled from one place to another. And if we find even one other example of life in our solar system, that strongly suggests it's all over the place, because we know so many solar systems are out there. Virtually every star's got one. And so, the chance of finding life in another solar system within our lifetime is fairly remote. I mean, obviously, unless there's an ET that, gives us a call and so on and says, hi, I'm here. But otherwise, I think our best chance is going to be within our solar system, and that's going to really raise the probability that life is all over the place. So what we've done is, now we've talked a little bit about what's life, and a little bit about the limits, and where you could map within those limits, at least within our own solar system. But what if they're tempting locations that you can't quite make? And so, for example, say an astronomer colleague comes to me and says, Lynn, you say that the highest temperature for life is 122. I found this place that would be fantastic, except that it goes up to 130, or it often hits minus 50. Do we need to scratch this off the list? And I said, don't cry, hand them a handkerchief, I'll make you something. And I really believe that synthetic biology has a role here, because we do know enough about these mechanisms to be able to start to push the optima. And in fact, our 2012 team did that as one of their other projects. They took about 15 or so genes and operons and put all of them into E. coli. And they didn't make, the monster E. coli, but they could show that very quickly, in the course of a summer, they could shift the niche space for even E. coli, so that you vastly broaden the places it could live. So imagine if we really put our minds to it, what we could do with synthetic extremophiles. I'm playing the synthetic extremophiles. The students have a much better name now. So I've used that name all the way up to the White House multiple times, and it is such a great name. I wish I still had it myself. But anyway, so that's sort of the, Cliff Notes looking for life in the universe. But then, about a dozen years ago, we had a center director. So we have, it's sort of like a university with different campuses. I'm at a center called Ames out in California, near Stanford University, not too far from San Francisco. And our center director at the time was ex-general, with an astronomer with a lot of vision. And he became really enthused by the potential for synthetic biology, particularly for human missions. And so he gave me a graduate student and asked me to start a program. Now, he was a 2 or 3-star general, so he actually didn't ask me, he told me. But nonetheless, we went merrily forward. And so rather than just, this is a picture from one of the TV shows I was in the Altiplano in Bolivia. Obviously, the sun doesn't look that big in Bolivia. Neither does Jupiter. But there was a new planet in the sky, synthetic biology. So I'm going to spend the rest of my time giving you a little bit of the flavor for what's going on in that neck of the woods, since I now wear two hats. So as humans go forth on planet Earth, which is also part of astrobiology, what's going to happen in the future, we're going to have very different sorts of things. So here is a top secret picture of NASA Ames, where I work. Ooh, two top secret pictures. The hangar that you see over there used to be covered until someone realized it had asbestos. Those were built for some of the dirigibles in the Second World War. So NASA actually was born in 1959, but we're one of the original NACA centers. So that was a precursor to NASA early in aviation. So we're actually about to become 80 years old. And I realize that's laughable at Cambridge. But 80 years old is very old. So as I said, NASA wasn't even born. We're only 50 years old as an agency. So anyway, this was a naval part of it. Naval aviation predates it. My lab is actually past that one-way sign a bit. And I probably should take a picture close up because it's kind of fun. It was built during the Apollo era, so the outside of the building has pockmarks to look like the moon. But anyway, that sort of starts you on planet Earth in California, and that's just one of those typical days out there, completely cloudless and so on, we're looking for rain and all that. And then you start to go up into space, and here we are. I've conveniently shown you the meatball so you can find out where NASA Ames is. There we are in San Francisco Bay. At the tip of the peninsula there is San Francisco itself. Berkeley's across the bay, and so on and so forth. If you're a wine aficionado, Napa and Sonoma, then would just be a little further north there. So you start to go up, and obviously you no longer see the buildings and so on, even if I hadn't put the meatball over them. And then you get to the moon, and Earth recedes into the distance. And you get to Mars, and you can barely see it. Can anyone here find Earth? There we go. And just because I just thought of this the other day, it would be really fun. It gets worse. I love this picture of Saturn, obviously. Can you find planet Earth? Can you find yourself in this picture? And there's Cambridge. Yeah. So the point of showing those is I think it very dramatically shows that as we move away from the Earth, particularly as we go to the moon and Mars and beyond, the Earth becomes this tiny little speck in the sky. It no longer dominates our lives. And therefore, we're not going to be able to be micromanaged by the people on planet Earth. It's not going to be, can I do this now? Can I do that now? Hello, Houston. You're going to have to be able to think on your own. And so I like to imagine that someday when we have more permanent settlements there, people are going to maybe be studying about planet Earth for their A-levels, or it's going to be this fantastic vacation that you take with your family. Where'd you go this year? Oh, I went to Earth. Or maybe it's your honeymoon. I don't know. But the point is that there may come a time that Earth doesn't even dominate our entire species. And so we're going to have to be able to deal with living apart from Earth. Now, when you're on planet Earth, this happens to be New York, there are all sorts of needs for human settlement. You need transportation. Couldn't quite get that in the picture. You're going to need life support. So you're going to need food. You're going to need oxygen, medicine, waste recycling, clothing, and so on. I often give talks to military groups, Department of Defense, and they say, oh, well, our problems are just as great as yours. And I say, are you kidding? You get gravity and oxygen. Harder. You're going to need habitats, of course. I mentioned radiation's nasty up there. You need power, heat. You need light. You need radiation protection. I actually thought of taking a picture out in Cambridge yesterday and substituting this in, and then I realized that it would be very difficult to make the argument about radiation protection yesterday. So you'll just have to take a look for it. And I'm going to add cash in there because the guy told me a lot of dollar bills. It isn't free to go to the moon or Mars, obviously. So you've got these needs for human settlement, but you also have a lot of challenges off planet. And one of the big challenges is upmass, or up volume. So any mass or volume that you're taking from the spacecraft is something that, first of all, has to be launched out of Earth's gravity well, and it also is taking up space or mass that could have been used for something else. So the number that I've been throwing around for years is it costs about $10,000 to launch a can of Coke into low Earth orbit. I was reminded recently by IAI, I think. I don't know if some of you are familiar. I wrote a little piece for them for Halloween. That I'm talking about a 16-ounce can of Coke and $10,000 worth of 9,000, 8,000 pounds, whatever. Anyway, you get sort of the idea that it's very expensive. And at that point, you have a can of Coke in low Earth orbit. You don't even have something on the Moon or Mars. The costs are probably coming down a bit with the commercial shuttles and so on. Having SpaceX and so on is helping, but it still is expensive, and it's always going to be expensive. So you want to be able to get around that. And so, as I say, that's tied very much into the cost of the mission. You're going to need to be able to store things. You can't suddenly say, oops, I forgot something or this broke, and I'll just go down to the store, call Amazon or whatever. You just can't get. I always say, you can't get overnight delivery in Amazon. No one ever laughs in England because apparently you can't hear either. You can't on the Moon or Mars, so maybe you'll recreate after that, I don't know. And obviously you need power as well. So you got to figure out, are you going to bring lots of radiation with you, or are you going to use solar panels or whatever. Now I would argue that the solution to many of these problems actually is a wonderful technology that we call life. So if you start thinking about it, life itself is programmable, it's reproducible, it's self-repairing. It can do many of these things that we need for life support. In fact, look around the room, this is a perfect lecture room to mention this. Virtually everything you see is biologically produced. It's amazing. And why not take advantage of that off-planet? And the other killer is, think about the mass problem. If you can have the cells divide off-planet, you solve the mass problem. You can't start with a little tiny spacecraft and have it divided and grow into a big adult spacecraft off-planet. But you can do that. Every one of you has already done that. Right? And we also have slightly different considerations. So some of you who are sort of in the biz may have heard the expression, oh, well, we'll just get sugar cane from Brazil. And that's often used as a feedstock in synthetic biology. So the last time I was in Brazil, I actually took a picture of a Brazilian sugarcane field so that you now can see what you're talking about there. Obviously, we're not going to have Brazilian sugarcane fields on Mars or the Moon, but you do have that on the Earth. And so when you start to look at issues with synthetic biology on the Earth, like what the environmental impact might be like, the ecosystems, or how you're disrupting different existing industries. So someone may be doing something that's almost as good, but you've got a whole industry built around putting petrol in your cars or whatever. You've got political stability problems, and this was brought home to me years ago when I talked about using transgenic cotton, and someone from the State Department said, we're not going there, because we get a lot of cotton from Indonesia, and keeping the Indonesian cotton farmers employed is important for stability there, and better, all these things that I don't think about because I don't have to, and of course the philosophical ones and so on. So when we get to the Moon and Mars, a lot of these issues change dramatically. We need to worry about the environment if we think there might be a life form there, because as you can tell, I'm an astrobiologist by heart. And if there was life on Mars and we destroyed it from being careless and lost our best chance to find our nearest neighbor, that would be an enormous scientific tragedy. So there are all sorts of planetary protection rules in place. There are Coast Guard treaties, Canadian space research. There are many countries that have signed on to this. We at NASA actually have a planetary protection officer whose motto is all the planets all the time. That great sort of Men in Blackish. But we do actually do that. Up mass is our economic problem. We don't have political problems at all because there are no cotton farmers there already, so we don't have to worry about it. We just have to worry about the philosophical issue, what if they're Martians? But we don't have to worry about that in the Moon because the Moon's dead and always has been. So this is, I told you, I think that biology is technology, particularly using synthetic biology, is going to be the answer. And so when I started pitching this, I was asked to go up in front of some people on the NASA advisory committee, and for some reason it was the aeronautics committee. And they looked exactly the way you might imagine them. They were all in white shirts and pencil protectors, and they don't all wear pencil protectors today, but they did at the time. And I did all this, great stuff, and I thought they'd think it was too wild and crazy. And instead, the chair of the committee said to me, "So, what's the big idea?" I thought I gave you the big idea, so I was like, okay, let's do Star Trek. So the rest of the talk's going to be sort of, what's the big idea? So let's start with something that we see around this room, that we use organisms for material production. So here's I swap out scenes from Scotland once a year. I think this one's outside Glasgow, but it's such a perfect one to show biomaterials. And there you've got the sheep that obviously are producing wool, and you could in fact get sheep leather or, sheep stomachs for your bagpipe innards, and all sorts of things, and obviously trees there and so on. So lots of great biomaterials. The problem is we're not going to take trees or sheep or silk, probably not silkworms, certainly not spiders, not cotton plants, not trees, and so on, off planet. We are going to take something like this. This is Bacillus subtilis. And you may notice there's some white dots in there. That's not a contamination. Bacillus subtilis is very good at making spores. And it turns out that the spores of Bacillus subtilis have been shown to be extremely hardy. They were actually flown outside of the long-duration exposure facility for nearly 6 years. Actually, the mission was designed to be much shorter. But it turns out to have been nearly 6 years. And it wasn't 100% survival, but it wasn't bad either. So these have actually proven themselves, and now my lab has data for about 3.5 years, which I'll show you coming up at the very end. So Bacillus subtilis is really hardy. Why not take Bacillus subtilis and just program them to be making the silk for you, or the wool, or the cellulose, or whatever, or I literally was at a meeting once at NASA headquarters when an engineer turned to me and said, "You think life is so great, could you ever program life to make rubber?" Like, excuse me. Rubber literally grows on trees. So that, why not? Why not get a bacterium to make rubber for you? So what would you do with these materials? Well, what about habitats off planet? So here's Apollo 11 behind me, and there's a lunar module. And I did a calculation. The volume of the lunar module, the habitat part, was the size of a cell block and Alcatraz C block if it was only 5 feet high. You don't even have to have been to Alcatraz, one of the highest security prisons in the world, to get an idea that these were not luxury quarters. So we're talking about a very tiny volume. And if you're the very first person to set foot on the moon, or even the second, third, fourth, fifth, or sixth, you can deal with that for a day or two. Fine, anything, this is really exciting. But for a long-term habitation, you wouldn't want to be in there. And so the proposals right now for the moon and Mars are to take the habitats with them, just the way we did during the Apollo era. And we're saying there's got to be a better way. Why not, for example, grow a habitat on Mars or the moon? So this is a drawing that was done by a colleague of mine, who's an architect that we work with, Chris Maurer, Red House Studios. And this particular structure would be made out of fungal mycelia. And this is one of the things that our adjunct team last year worked on. So you're all familiar with a mushroom, and there's the fruiting body of the mushroom. You may be less familiar with what goes on beneath the surface, and that's these thread-like structures called mycelium. That's actually the fungus. So the fruiting body that you eat is just the fruiting body. So if you take this mycelium, and there's a micrograph of it, and you inoculate a mold with a mold, and put in grass clippings, which you wouldn't find on Mars, or sawdust, or, basically garbage. And we've done this in my lab with everything from used coffee grounds on, you name it, they will grow. And they will grow to fill the mold, and at that point you can bake them or just dry them or whatever. And in fact, we've even used that kind of approach to make the body of the biodegradable drone. So there you see the fungal mycelia, and then eventually it was covered with nanocellulose and so on and so forth. But let's get back to this mycelium. One of the questions we asked last year is, could you then grow it, say, on a Mars Regulus simulant? We don't actually have good surface material from Mars. The meteorites have all been melted and so on. But we do have what we think is a good simulant, and you can see that mycelium growing on the plate. You can take things like this, as I say, make structures, and actually fairly quickly. So here is a little stool. It's not particularly attractive, but it's one that the students made last summer, and you can see it's rated for one average Stanford student. So these are very easy to make. Why not actually grow an entire habitat or a table or whatever? In fact, I have a grad student in the International Space University as an architect working on that right now this week. So, there's a great idea. You could grow a structure there. You wouldn't have to worry that the ground was absolutely level or anything like that, because you could get the mycotexture to fill in the cracks. Now, obviously, I'm not suggesting just, spitting out spores. What we're suggesting is having these in a double bag sort of situation, so you can keep the pressure a little bit higher. You can keep some moisture in there and then let them grow to fill the bag. Another big idea is sending digital information. And this, when you hear what I'm talking about, it's sort of like, oh yeah, that's so obvious. But it hasn't been to a lot of people. Normally, if you take something with you, you need to take the physical, physical object with you. But for something like DNA, and I'm sure many of you in the room have this sort of experience, you go to your desk, you come up with some sort of sequence that you want made, and you hit send, and your supervisor pulls out their wallet, and next thing you know, there is some DNA synthesis company making that for you, and then sending the physical DNA back to your lab. It doesn't matter whether that DNA synthesis company is across the street, across the continent, or across the solar system, because once you have digital information, we can send that anywhere. So you've now broken that physical barrier, so we can come up with new cures or new ideas for how to program organisms, send the digital information off planet, and then have a DNA synthesizer there to actually make it. So that's completely massless now, because you've got a single system that's being able to do all sorts of things. Biology does chemistry on the Earth, and I could write fancy equations on the board, but no one wants to see them, so I'll just show you this picture. We're going to rely on nanotechnology, and when you hear nanotechnology, your immediate thought is probably clean rooms and all sorts of fancy equipment. But what if we could take advantage of biology? That's crazy. Now, I'm not talking about taking advantage of a graduate student or a postdoc. I'm talking about taking advantage of what actually goes on in your cells. And so years ago, I ended up being the opening speaker for a nanotechnology conference, mostly because the first speaker got tied up in LA traffic. But anyway, there I was, and the night before, I'm thinking, why did I say yes to this? I know nothing about nanotechnology, and here are all these prestigious scientists and engineers, and then I had an epiphany. It's like, I'm a biologist. The best nanotechnologists in the world are every one of your cells. I mean, if we don't have the option of saying, oh yeah, for our proteins, so we've got like a 15% error, and some of them are this long, and some of them are... That doesn't work. If you're going to be alive, you better have atomic and molecular scale precision assembly. So why not take advantage of that off planet? So for example, I've had a student who just got his PhD and continuing to work together, who had an insight that if you, which actually you heard about somewhere else, but if you have a cytosine-cytosine mismatch in DNA, it turns out silver atoms will uniquely go into this mismatch. And so what you could do, therefore, is electrify DNA. So you just, you do a whole string of cytosine-cytosine mismatches and do a GC plant at the end, every other one, 3 in a gap. You can do all sorts of things. And then he went on by the end of his PhD into starting to make 2 and 3D structures. He said you're not supposed to use the word DNA origami, but it's sort of like electrified DNA origami. So very, very cool stuff that we're continuing to push ahead with. What about metals? Metals have a lot of mass, and there's plenty of it off planet. So what about just taking the metals that are there? Similarly, what about recycling spent electronics, whether you're on the Earth or off the Earth? So I always tell people the best place to find the ingredients for a live cell phone is from a dead cell phone. If you just had a way to recycle them, and it takes zero effort Googling to find out how that's done on planet Earth today. You will see horrific pictures of people bent over fires in places like India with fry pans and things like cell phones. This is not good. This is not sustainable. We need a better way to do it. So again, let's turn to life. Now, many of you probably don't think about this every day, but we're organometallo compounds. Many of our protein, though 50% or so, use metals at some point. So we have the ability to use and take metals. And so people ask me, what metals? And I say, look at the back of a vitamin bottle. That's a really easy way to sort of get a quick idea. It's not everything, but it gives you an idea. So what if we take advantage of the fact that we already know that there are peptides that bind metals and use that as a way to extract metals, say, from spent electronics? So we've been working on this since 2012. iGEM team, Jessica now has her PhD working on it. We have a paper that came out in Scientific Reports a few weeks ago and a patent disclosure. Actually, it's a provisional patent that's come out on this. And the idea here is that we could grind up used electronics. We engineer these peptides to bind metals, and then we simply take those and feed them into a plasma printer, and you can reprint interconnects that actually are functional. So we did this with copper as a proof of concept, which gives you the ability to do the interconnects to test. But what if we now combine that with the microtexture? And this is actually what was in the patent. So we no longer worry about maybe expressing these metal binding peptides on the outside of flagella, but instead put a chitin binding domain on the other end and attach them to the microtexture. And as I told you, fungi are not picky eaters. You can literally feed them garbage, so your cost comes down to practically zero for the substrate. So you really could make a Brita filter sort of thing with this. So I say we've got this provisional patent out, and we now have some money from the U.S. Geological Survey and various other people trying to get interested, because metals are a huge problem, and access to rare earth metals is going to be a huge, not just problem for the high-tech industry, but it's going to be a geopolitical and a military and an economic problem over the next few years if we don't have other ways to do this. Generating electricity. Yes, you can generate electricity from chemical energy. Every one of you is still doing this today. That's how our brains work. It's like the eel's got a master switch to be able to release all at once. And so there should be a way to take this ability to create a charge and release, store and release. And I have a former student who's working on his PhD doing that right now. Why not this life 1.5? Why not create these health cells that therefore can save you power? So you've got organisms that might grow at 37 degrees on the Earth. You bring down their optimum so it's closer to the surface of Mars. And so that saves you power you might need to keep. And by the way, that's Yellowstone National Park. Has anyone here ever been to Yellowstone? Okay, so anyone here hear about Taq polymerase, PCR? Yeah, a lot of heads nodding. You're too tired to put your arms up. So this is a little bit of trivia. This is Octopus Springs in Yellowstone, and if you tilt your head sideways, it does sort of look like an octopus. You see the tentacles coming out. And the main body is above the boiling temperature of water, that beautiful sort of blue turquoisey. But then you can see the outflows start to be different colors as the water cools a bit. There's my daughter, who is now working on her PhD, so you can tell this is a very old picture. But what's kind of fun is this is Octopus Springs, but about a 5-minute or so walk from there is a spring that looks kind of like that, with the hot part and the colored bits that looks more like a mushroom. And interestingly enough, it's called Mushroom Spring. And that's where Thymus aquatus, and therefore Taq polymerase, was first isolated from. So now you sort of have an idea. It was from going out and sort of mushing around, and I can tell you that story sometime. Well, why not make life de novo? Why not make something that's just, happier on Mars or Titan or the Moon or wherever? Again, maybe you don't need to have squishy lipid double layer membranes. Maybe you could do something utterly different, a protein membrane or a silicate one, or who knows. And so there are actually people in the Netherlands, I know, there are people in, we've got a network going in the US that we're expanding beyond called Build a Cell. There's going to be a little meeting, we meet about every 6 months. I'm soon to be hosting the next one, so I hear. Anyway, so there are people who are sort of going that direction, mostly for synthetic biology applications. Obviously, I have an interest in it for origin of life as well. So it should be interesting. And I should say that one of my colleagues who's been heavily involved in this sort of research building cells for many years, Dave Deemer, came to speak to my class once in Stanford, and I want to tell you a story because I think it's really interesting. Dave is a reasonably modest person, just turned 80, and I asked him in front of my students whether he thought that he would ever make life, or anyone, within his lifetime. And he says something very interesting. He said, I may have done it and flushed it down the sink. So what are you talking about? He said, life is a combinatorial chemistry problem, and you can't interrogate everything that you've ever made. So maybe someone has already made life and flushed it down the sink, which I think is a rather intriguing idea. And there's a little organism making life. Gives me a chance to. And if you're going to ask the question how you do that with PowerPoint, the answer is you don't. You do that with Keynote. Okay, so what happens when an astronaut gets sick? It's not totally worthless tonight. And so one thing we tackled last summer with the iGEMers is making what we called an astropharmacy. And I would like to think that we came up with that name, but a colleague of ours, Phil Williams at the University of Nottingham, was the one who's actually in the pharmacy department. He said, I think you should make an astropharmacy. And the name was so great, we just had to do it, even though I knew absolutely nothing about pharmaceuticals. But it turns out that biologics, protein-based pharmaceuticals, are getting to be a big thing. And it turns out that astronauts have several hundred medications they take with them. But the problem is you don't know everything you're going to need, and even worse, over periods of time, and certainly with space radiation, the efficacy goes way down. So even if you could take every medication known to mankind, many of these things, particularly the protein-based ones, would go bad in 6 months, even with refrigeration. And if it's about a 2.5-year round trip to Mars, that means they're gone. So what we tackled was the problem that's the exact opposite of pharmaceutical companies who want mass market, make lots of money. We asked the opposite question: what about in 24 hours making a very limited number of doses, but keeping the precursors at room temperature? And so we built a little protein-based system for tetraparatide and PSSF, and there's a little prototype, and we're obviously getting some interest from NASA at this point, and that was that wonderful group of students. So let me finish by saying that people then say, well, these are, this is all great sometime in the future, blah, 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 blah. What about actually testing this? Will any of this work in space? And just because I've been at NASA since before many of you were born does not mean that people knock on my door every day and say, hi, how would you like to send something to space? But I did finally get a call about 5 years ago or so, this is 2012, a little longer, from our chief scientist who was with our center director at the German Space Agency. And he called and said, we're talking to the head of the German Space Agency Monday, and we think they're going to offer us a secondary payload on a satellite mission. Do you have anything, preferably synthetic biology? It's like, oh, sure, I'll just, this is not the way this normally works. Normally you have 3 years of funding to develop an idea and so on. This was like, you've got 48 hours to flush out a payload. But you don't turn down an opportunity to go to space. So I did. And it turns out there were 3 of us, and then there were 2 of us, and then there was me. And this is what the result is. It was the PowerCell payload on Eucropus. PowerCell was one of those 2 iGEM projects that very first year in 2011. The idea was to be a biological nerve center for a synthetic biology-enabled colony. So you're going to start off with a cyanobacterium, which can fix both CO2, or it's diisotrophic, also fix nitrogen, and use those as the organic inputs to be able to feed your Bacillus subtilis or whatever that's then going to make your wool and your spider silk and your habitats and feed your fungi and all that. The reason this is a particularly interesting satellite in Crocus is, which stands for Lynn across in space, is the idea is that it's going to spin at 3 different gravity regimes. So first we have a chance to test these experiments at microgravity, and then a test at lunar gravity, which is a 1/6 Earth's gravity, and then Martian gravity, which is a 1/3 of Earth's gravity. So you really can run a controlled experiment sequentially in the same spacecraft. Obviously it needs to be balanced, so we had 2 flight cards in 2 different payloads on either side, so there are 4, so obviously we could do one twice. Well, the idea of that power cell was to just really replicate what you see on the Earth. You've got primary producers taking the sun and CO2 and the water, so basically interfacing between the raw materials on a planet and then things like cows that would then, or sheep, make wool and so on. And the idea is you swap out microbes to do these things, and then you take them out of Bolivia and you take them off planet Earth and you dump them in someplace like Mars. So to me, it's not a very radical idea. It was more radical to my colleagues who think you're going to do this all, excuse the expression, with chemistry. You might as well take advantage of life, because life did a very good job of interfacing on planet Earth. Why not do it off there? And since flight experiments are very expensive, and I had basically the form factor of a 96 well plate to work with, we decided we could not do even more than power cell, that we could also test some other synthetic biology principles, like do a transformation at different gravity regimes and look at promoter strength. So transformation, how many people here have done transformation before? So you may have used something like electroporation. If you want to watch an engineer turn pale, say electroporation, or just say, we want this jolt of electricity. So that was definitely a no-go. And so how do you make something like bacillus competent without using electricity, just sort of on command, when you've got fluidics and temperature basically as your possibilities to play around with? And so this lesson to those of you who are biologists in the room, you call a culture collection. And a really good curator on a culture collection is going to know everything. And the way I always describe it, because it just reminds me of it, it's sort of like the scene in Harry Potter where he first gets a wand. And he goes to Ollivander and he picks the wrong one, and then he says, wait, I know just what you need. And he goes in the back, rummages around, and finds the right wand. That's what a good curator does in a culture collection. You say, I need a bacillus subtilis that's going to become competent when you just add something that's not toxic. And you say, I've got just the one for you. So it turns out this one becomes competent with 2% xylose. So what we were able to do is add culture medium with 2% xylose to activate that particular experiment. I won't go through the details of the other things because we don't have time for this. You've already been here longer than we were planning to, I'm sure. Here is one of our flight cards. Each one, there were 48 wells, 4 rows of 12, and each one of those little wells basically had a spectrophotometer in it. So what I could do is fluid flow and look at the optical density and again temperature control. We loaded these samples May of 2016, and then we waited. And we waited, and we waited, and I started to get increasingly nervous, like I was going to be hung out to dry if these things died. And so then I started begging for a reload, and I was told it's too expensive because I hadn't realized that they were going to build the hardware around the samples, so they would have had to dismantle everything, which I think is insane. And it's to cut down the mass, but in the future we're really trying to push having something more like lab equipment where you stick a sample in. Anyway, we waited. So May of 2016, the samples were loaded. We finally launched December of 2018. Total shock that this finally went off. And here's a movie. I don't know if it's playing or not. Anyway, look by the Brazilian film crew. It's the sort of movie that only a mother could love, sort of thing. Anyway, we went up, and SpaceX launched it, and they dropped a bunch of things off, and we were one of them. And then, once the system's checked out, we finally loaded the first card. It filled nicely with the fluidics. They're the engineers, and they're me, with the our cheap champagne, great excitement. Finally, this thing was going to work. I was hoping it was going to work, but at least the fluid flowed. So they said, well, that's like half the battle. And then, to those of you who follow the newspaper, the next thing that happened is we went on furlough. So I was one of the government employees who was allowed to work without pay, very fortunately, she says somewhat sarcastically. But anyway, because of that, we actually let the experiments go a lot longer than we would have normally because we had nothing else to do except call each other every 2 days to see how it was going. So yeah, really cool. And actually, this is really unfortunate. The Germans still now, almost a year later, have not been able to establish contact with their primary payload. So we have been the science output. And I am pleased to say, as of, well, this is October, but it's now November, we've been able to activate the microgravity, run that, they say literally for months. We only run it for 10 days, we ran it for 83 days. That's, that's furlough absurdity. And then the ground controls, you do the ground controls offset about 10 days, just in case there was a temperature spike or something that you want to mimic on the Earth that was unforeseen. We've also done the lunar gravity, and we've done Martian. So we've done everything once now, and we should have one more payload to go, but we're still waiting on the Germans to tell us that hopefully we can do it, that they've reestablished contact or whatever. Fortunately, I'm going to be seeing the head of the German Space Agency Friday, so hopefully there's some good news. But let me give you a couple of highlights. We now have over 400 days worth of mission data, and as I was saying sort of in the middle of the talk, Bacillus subtilis went on LDEF. I flew Bacillus subtilis not because I love them. I have many more favorite organisms, but because it has allowed me to sleep at night for the last 5 years. And by golly, the spores came through. You add water and they came through. We've noticed growth in all the gravity regimes. We've had a successful transformation. Oh, so let me show you a little sort of data. So this is from just ground controls. And what you do is you dry Bacillus subtilis spores, and then every now and again you hydrate them and plate it out and see how many are still around. And you can see we've gone down in order of magnitude. You can see these are started at different concentrations and they're different strains. But basically, you go down in order of magnitude and then it pretty much levels off. So it's been really, really a godsend. There's no other way to put it. We were able to do the power cell. Now, we weren't so secure about being able to have that kind of stasis with the cyanobacteria. In principle, they should have. The one we used should have been able to form resistant spores. But we weren't able to have enough confidence, and I'm really glad we didn't because we weren't anticipating 3 years of sitting in a flight card. And so that would have been a nightmare. So what one of my students suggested is that we cheat a little bit and that we grow up the cyanobacteria and then break them open and send them lysate. And that's what we did, and they grew beautifully on that. So that was great. It doesn't look like much because we didn't blow up the axes, I should have, but the long and short is all the strains grew on that. So that was fine, and it was pretty much the same in the 2 different gravity regimes. The transformation of microgravity worked, which I could not be more thrilled. This is actually, raw data, and what you see is in the black are the empty wells. So we've got some of the wells that we didn't put DNA or cells in it, and what you often do get are air bubbles coming into this microfluidic system in space missions, and then you start to see the controls go all over the place. But here we didn't get any air bubbles. It's just picture perfect. We have a parental strain that was sensitive to the antibiotics we put through neomycin. We had the orange one we transformed with the neomycin resistance before we sent it up. And then the transforming strain had the DNA for the neomycin resistance, but it was a sensitive strain when we sent it up. So the only way it could have had that kind of growth curve is if it transformed. So just thrilled about that. The unfortunate thing is that the growth on LB also worked in the microgravity. This worked, and then the subsequent ones got worse and worse. And it turns out it was not our bacillus that let us down, but from the ground controls, we discovered that if you keep 2% xylose in LB, it turns brown eventually from probably Maillard reactions. And so you can see LB is supposed to look like that straw color went all the way to the left, and then over time it starts to turn dark brown. And if all you've got is OD to look at, that's a problem. And it turns out that in spite of the fact the American Chemical Society says that we love the byproducts of Maillard reaction, the psilocybins don't. And so they just wouldn't eat it. So lesson learned, and if we'd been able to have all this time in advance, we would have known that. So that's why some of these subsequent ones that are not working, it has nothing to do with the gravity, it has nothing to do with the psilocybins, it has everything to do with the Maillard reactions. So we've got one more fluidics card to go, and even if they won't let us activate that, we've got one more ground control. So that's sort of the message. It took you from astrobiology to synthetic biology to actually get into space. So to leave you sort of with some final thoughts, we have all these needs for human settlement. I do firmly believe that biology is going to be the answer, but this is very different from the sort of questions and approaches that most of my colleagues in synthetic biology have, but I think because we have these constraints, it means that we can be more creative. I think I finally described it at the meeting at the White House 2 weeks ago. You know the old joke about Fred Astaire, being the world's greatest dancer? But the response was that, yeah, but Ginger Rogers had to do everything he did except backwards and high heels. And I feel like we're sort of the Ginger Rogers of the synthetic biology world. We have to do everything everyone else does, but we have to do it with no oxygen, low mass, and, altered gravity. But this means that we can come up with creative solutions that I firmly will believe, will force game-changing solutions that will help planet Earth as well. PowerCell is the first start on that. And so, to sum it up, we've done all sorts of things in my lab over the years. It's been a fun ride. I tell my students, I've gone as high as I can in the government. I can't get promoted anymore, so you might as well have a good time and do great things. And so that's what we do, whether it's astrobiology or synthetic biology or some combination. And so I end with ad astro, which means to the stars. Thank you very much.
I was really curious actually about what you said about finding a second example of life within our own solar system. How would you really know that it's a second example of life and not just life that used to be on Earth, or maybe our Earth life used to be on Mars,
right? And
then we don't really know how common or rare life is elsewhere in the universe.
Right, and that's a really difficult thing. I'm not convinced, John Sutherland's recent work aside, that the first thing that you would necessarily see is DNA or RNA. I would be more suspicious that you would get proteins. There are about 10 amino acids that we use that are commonly made abiotically, and then the other 10 are only made biotically. So you can start to play games like, if they're using the same ones that are only made biologically on the Earth, that's sort of suspicious. I personally find DNA suspicious. One of the common things that people talk about is looking at chirality. Of course, we all use left-handed amino acids and right-handed sugars. So any deviation from that could be indicative of a second genesis. So those are some of the sorts of things that people look at. I really have very little patience with my colleagues who say, oh, well, we'll just find a Martian, we'll sequence its DNA and see if it's on the, our tree of life. It's like, if you're that far, it probably is. So anyway, that's sort of a quick answer.
More questions?
Yeah, I was just wondering about kind of like what you were talking about with using engineered, bacteria probably or other like stuff for space exploration. So how do you ensure that your engineered organisms can actually like stay stable throughout long periods of time? Because obviously, like with time, you can have your engineered constructs being selected out and all that kind of stuff. How do you prevent that from happening if you've sent your bacteria all the way to Mars?
Right. So again, that's why I am so bullish on Bacillus subtilis, our few other bacilli, because you literally can dry them like we've done, and by the time you dry 1,000 or even 1,000,000 spores, there's almost no mass. They're not going anywhere. And so you could just keep re-inoculating with tiny bits of spores and not even worry about it. I mean, you literally could do that. Why not? Whereas you don't have that, you're right, that same sort of option if you're going to worry about continuous culture.
Did you try to clone some DNA samples obtained from Moon or Venus? And if you clone, you use Earth condition or Moon or Venus condition?
We've never found DNA from the moon.
Okay.
It'd be very cool.
Given
In
a
fact,
report a while that they
No, I mean,
found a
it's-
bacteria.
yeah, no, we've never found anything like that. Believe me, this would have been a very different talk. If you think I'm excited now, you should think. I will say that that's slightly disingenuous because obviously we've had people on the moon. And also when we spend spacecraft, we try to, we don't actually sterilize them, we reduce their biology. Again, can you imagine building this advanced spacecraft with all sorts of electronics and then inform the engineers you're going to put the whole thing in the autoclave? It just doesn't, that doesn't go. And so we reduce the bioburn. So there has been a rumor out for a long time that when the Surveyor camera was brought back to the surface of the moon, there were microbes that had survived. And that's an urban legend, so I'm going to disabuse you. Our former planetary protection officer has looked very carefully at the tapes that were taken when they opened up the camera on the Earth, and he is sure that he has found a time where they contaminated the samples, the camera, before they looked for the microbes, and so he's sure that it wasn't done. But I know there are people who are still very interested in going to, for example, the leftover lunar module, and I didn't realize that this was a thing until the other day. Someone said, well, there are astronaut diapers on the moon. It's like, there are. So apparently there are astronaut diapers on the moon, which would be really interesting to check, but I suspect there's nothing that's still alive there. Yeah, how do you top that, right?
What do you think about looking for biosignatures in exoplanets?
I would love that. We were just talking about that at lunch yesterday. And so one of the things that Jim Lovelock, who's just turned, 100, had suggested, I honestly didn't know I see still alive. Martin Rees yesterday showed me a picture of his 100th birthday party. Whoa. Anyway, what he suggested years ago, and I believe Lynn was making part of it too, is looking for atmospheric disequilibrium. So we have a situation of 21% oxygen and 79% or so nitrogen, which is an unstable condition. And so the idea is that to create this disequilibrium, we would have needed organisms. I think there are probably other ways, but that's been sort of one possibility. Looking for water in an atmosphere, helps. Obviously I would love to see some sort of organic compounds in there. There are people who talk about looking for the red edge, looking for something that would mimic chlorophyll, which of course is critical to life on Earth, but there are other ways to capture light energy, and there was life before there was photosynthesis. So that would be really exciting if we found something like that. It may or may not be proof positive, and it may not even be necessary. So these are really interesting questions. I've had astronomy students in my lab doing catalogs of color, and it's pretty hard to find any color that life on Earth hasn't figured out how to make. So, it's a really, really interesting question, and obviously that's something that I'm very interested in. What can you possibly get from atmospheric spectra? We got to pushing for lunar stuff as well, because if we're going to start worrying about Europa and so on,